Hi there, I'm Margaret Roden and I am presenting an article on giant viral signatures on the Greenland ice sheet. This study was set up to demonstrate that nucleocytoplasmic large DNA viruses live in the extreme conditions of the Greenland ice sheet. And it also asks, if they do live in these extreme conditions, are they present or infect snow algae? This could potentially be used as a biological control for algal blooms that contribute to ice sheet and glacier melt. So a little bit of background about the Greenland ice sheet and glacier melt. The Greenland ice sheet forms the second largest body of ice in the world. It averages a thickness of about 1 mile with a maximum thickness of 1.9 miles. It is approximately 1,800 miles long and 680 miles wide. Some of the factors that contribute to Greenland ice melt are an increased melt due to warmer air temperatures, melting of ice along the sheet margins, ice calving into the sea from glaciers and the sheet edges, dark ice increasing temperature of surface ice, blooms of algae and bacteria growing in increased numbers furthers the melt. This increased ice melt has major global impacts on plants, animals, weather, commerce, and coastal erosion, which all affect global food supply. This study explores 5 types of ice. Cryoconite, which is dark-colored, spherical, organic matter aggregates that are light-absorbing. This light-absorbing causes the surrounding ice to melt and form a hole with water, allowing organic growth. Green snow, which is snow colored by the growth of green algae. Red snow, snow colored by the growth of green algae with a secondary red carotenoid pigment or with red algae. Ice core is simply a core sample removed from an ice sheet or glacier. And dark ice, an ice sheet or glacier coated with cryoconite. So what are nucleocytoplasmic large DNA viruses, or NCLDVs, which we're going to refer to them as for the rest of this presentation? They are double-stranded DNA viruses, and they are a diverse group of viruses that can infect metazoans, algae, and heterotrophic unicellular eukaryotes. Their virion size can reach up to 1.5 micrometers, and their genome ranges from below 100 kilobase pairs to more than 2.5 megabase pairs. NCLDVs provide biological control on algae and marine freshwater environments, but little is known about their distribution and function in terrestrial ice. So a little about where these samples were obtained. During 2 fieldwork campaigns in July 2019 and July 2020, 19 samples were obtained from the Greenland ice sheet. 1 sample was collected from cryoconite. 3 samples were ice core samples. 2 samples were collected from green snow. 5 samples were collected from red snow. And 8 samples were collected from dark ice. In addition to these samples, researchers also included the analysis of 1 metavirome, less than 2 micrometer fraction, from the dark ice. Although it's not stated in the paper, when researching the paper, I found that these sample locations corresponded to increased melting rates on the Greenland ice sheet. In 2019, samples were collected from 3 locations on the southeast of the Greenland ice sheet. Location 1 was on the Miktivakt Glacier, an independent glacier separated from the Greenland ice sheet on the Ammassalik Island. Location 2 was on the Greenland ice sheet across the fjord from the Miktivakt Glacier. The 3rd location was on the Buckner and Heim glaciers. In 2020, samples were collected from the Greenland ice sheet close to the QAS-U and QAS-M Promise stations in South Greenland. Field samples were collected using sterile nitrile gloves and tools and stored in sterile Whirl-Pak bags. Samples were melted at ambient temperature in the field and filtered through 0.22 micrometer mixed cellulose ester membrane filters. The samples were immediately frozen and shipped using liquid nitrogen and stored at -80 degrees C until analyzed. Total DNA and RNA were extracted from the filters with the PowerLyzer PowerSoil DNA isolation kit and the RNeasy PowerSoil Total RNA kit, following the manufacturer's instructions. 19 DNA libraries were generated with the NEB Next Ultra 2 FS DNA library prep kit with 8 rounds of PCR amplification. Sequencing was performed using the NextSeq 500 platform and the 300 cycle version 2.5 chemistry. These had 151 base pair-in reads. The environmental transcripts reconstituted full-length ribosomal RNA small subunit genes were taxonomically identified with SILVA 138.1, BLAST, and CREST4. Chloroplast and mitochondrial sequences were removed, and the assembly diversity statistical comparisons were analyzed with PhyloSeq version 1.44 and RStudio version 3.17. For the snow algal genomic amplification and assembly, the algal strains Microglena CFSP 002B99, Chloromonas ramaceae 047-99 and 005-99 CF, and Sphaerocytis species 101-99, and Raphidionema sempervirens 011A-99. These are commonly present on pigmented snow and surface ice of the Greenland ice sheet and other glaciers, and were obtained from the culture collection of cryophilic algae at the Fraunhofer IZIBB Institute. They were grown at 10 degrees C in liquid triple concentrated Bold's basal medium, pH 5.5, under anexic conditions and continuous illumination as per the CC Cryo guidelines. DNA was extracted using the PowerSoil DNA isolation kit following the manufacturer's instructions. Each strain was sequenced on a MiSeq flow cell using the 500 cycles version 2 chemistry 250 base pair-in reads at the Genome Analysis Center, Earlham Institute in the UK. In addition, high molecular weight DNA of Sphaerocytosis species 101-99 was extracted using the QIAGEN Genomic Tips Extraction Kit and sequenced using PacBio Sequel SMRT Cell 2.0 chemistry at NERC Biomolecular Analysis Facility in Liverpool. The genomes of all 5 strains were de novo assembled by the Earlham Institute using Illumina 250 base paired in reads. Quality control of the raw data was done using FastQC. Pre-processing of the raw reads was done by the Earlham Institute using the Pipeline Contaminant Abyss version 1.90, and it was used to perform the de novo assembly of each strain. The PacBio Sequel reads of the strain 101-99 were de novo assembled using Fly version 2.3.3, using a minimum subread length of 5,000 base pairs and an estimated genome size of 120 megabase. Sample MG32 was extracted with the CTAB citrimethyl ammonium bromide method and sequenced on 2 platforms. Illumina libraries were prepared using NebNext Ultra 2 fsDNA library prep kit and sequenced on a NextSeq 500 instrument with the 300 cycles version 2.5 chemistry. Nanopore libraries were prepared using the ligation sequencing kit LSK 109 and sequenced on a MinION with FlowMin 106 flow cell. Controlled using MinKnow 19.10.1. Raw nanopore Fast5 reads were base called with GPU Guppy 3.2.6 AFC8E14. The Illumina reads were quality filtered using Trim Galore under default settings. The raw nanopore reads were corrected with the trimmed Illumina reads using Lordic with default settings. The corrected long reads were used for de novo whole genome assemblies with Fly under default settings utilizing the Nano Core flag. The dark ice environmental sample MG32 containing a high abundance of Ancylonema species was taken in an attempt of producing a draft genome of the Streptophyta glacier ice algae. The overall appearance of the sample under the microscope gave the misleading impression that this mixed culture would contain primarily Ancylonema species and prokaryotes. The idea was then to remove the prokaryotic contigs and have a representative Ancylonema genome. Further analysis on the resulting assembly with BARNAP, Basic Rapid Ribosomal RNA Predictor, revealed the presence of a diverse eukaryotic community. Nevertheless, they chose to include this sample in the study as it provided another dark ice environmental sample and gave nanopore long reads. Metagenome, metatranscriptome, and GVMag assembly. Illumina reeds were quality filtered to remove low quality reeds and trimmed with FAST-P version 0.20.0 using default options. Trimmed Illumina reeds were assembled with Meta Spades version 3.15.1, specifying the only assembler pipeline. Metatranscriptome reeds were quality cleaned and trimmed with Trim Galore version 0.6.6 using default options. Raw reeds were assembled both singularly and pooled with Trinity Assembler version 2.6.6, including the following options. Normalized by read set. Giant virus metagenome assembled genomes, or GVMAGs, were created by binning contigs with MetaBat2 version 2.12.1 using greater than 5,000 base pair contigs. Resulting bins were analyzed for NCLDV marker genes using viral recall version 2 and were considered a GVMAG if they had 5 or more of the marker genes, a genome larger than 100 kilobase pairs, and taxonomic placement within other NCLDV genomes. COVER-M version 0.61 was used to assess the read recruitment between all 57 generated assemblies and the environmental sample reads. GMAG's functional annotations were assessed with InterPro and GVOGs. For the Metavirome assembly, 5 liters of dark ice from the Greenland Ice Sheet 2020 location was pre-filtered with 3 micrometer nitrocellulose membrane filters to remove large particles and subsequently filtered through 0.2 micrometer vacuum cap devices, retaining the viral fraction less than 2 micrometers. Viruses were further concentrated from filtrate using iron chloride flocculation followed by storage at 4 degrees C. After resuspension in a ascorbic EDTA buffer, pH 6, viral particles were concentrated using Amicon Ultra 100 KD centrifugal devices from Millipore and extracted as previously described. Briefly, viral particle suspensions were treated with Wizard polymerase chain reaction preps DNA purification resin at a ratio of 1 ml sample to 1 ml resin and eluted with TE buffer using Wizard mini columns. The DNA library was prepared following the Nebnex Ultra 2 FSDNA library prep kit with 8 rounds of PCR amplification. Sequencing was performed in-house using the NextSeq 500 platform and the 300 cycle version 2.5 chemistry, 151 base pair N reads. The sample was processed with a small filter size, less than 2 micrometers, and treated with environmental viral fraction. And it's important to note that the small filter size will decrease the amount of NCLDV signatures retrieved. Identification of the NCLDV signatures in the metagenomic data. Viral recall was used to identify NCLDV-like sequences and viral-like regions in all the metagenome, metatranscriptomes, metavirome, and pure algal culture. Options were as follows. DB marker C. The marker option was used only to search against 10 NCLDV marker genes encoding for factors for maturation of the viral cap. SID MCPs, packaging ATPase, A32, DNA polymerase elongation subunit family B, Pol B, D5 like helicase primase, D5, mRNA capping enzyme, mRNA-C, RNA polymerase large and small subunit, RNA-PL and RNA-PS, DNA or RNA helicases of superfamily 2, RNR and SF2, and poxvirus late transcription factor VLTF3 like VLTF3. All resulting hits with an E value less than e to the negative 10 were used further. These genes are universal NCLDV marker genes and hence are routinely assessed for identification of signatures of NCLDV in different ecosystems. Pol B is the only marker gene typically found as a single copy and is therefore used for phylogenetic placement within known NCLDV families. To confirm that virus-like regions belong to the NCLDV families, BLAST-P function against NCBI was used, and 50 top hits were verified for each sequence classified as possible NCLDV gene by viral recall. A gene was considered from NCLDV when it had NCLDV results within the top 10 hits. The total abundance of the 10 NCLDV core genes in the sample was calculated before and after verification with NCBI by summating the marker genes with an E-value cutoff of 1 times 10 to the negative 10 and normalizing to the total library size. 4 of the 19 environmental samples with the highest relative presence of viral marker genes was chosen to be resequenced at a greater depth to provide higher sequencing coverage and increase the chances of assembling GVMAGs. So for the results, giant virus marker genes were present in almost all of the samples. The highest abundance were recovered from red snow and snow algae genomic assemblies, followed by green snow and dark ice. Metagenomic data from red and green snow contained evidence of giant virus metagenome assembled genomes from the orders Imidavirilis, Asfuvirilis, Algovirilis, and metagenomic evidence reveals diverse NCLDV signature genes in snow and ice habitats, indicating the potential of viral controls on these algal communities. Researchers compared marker genes against the NCBI non-redundant database and removed fragmented hits to reduce false positive NCLDV marker gene identification, which increased the quality of the remaining genes used for phylogenetic comparison. Inconclusive matches occurred with all tested marker genes due to the presence of hypothetical proteins generated from poorly annotated bacterial mags and unknown viruses within eukaryotic genomes in the database, resulting in 879 marker genes. To determine the speculative total of the number of NCLDVs present, membrane cofactor protein genes were summed and used as proxy for the number of NCLDVs. These MCP genes are considered bona fide viral genes, and there were 211 MCP genes as a result. Quality control measures were applied to the MCP genes, leaving only 67 MCP genes remaining. The quality control measures potentially limited the detection of novel NCLDVs, but also demonstrated the potential diversity and abundance of NCLDVs in the Greenland ice. The 19 environmental metagenome libraries varied in coverage and size, but did not relate to the number of NCLDV marker genes identified. NCLDV genes were not found in 3 of the ice cores, or 4 out of the 9 dark ice metagenomes. The D5, RNA-PL, and RNA-PS genes were the most abundant in all the metagenome samples, making up more than 50% of the marker genes. RNA-PL and RNA-PS marker genes in the pooled metatranscriptomics had the most individual counts of the transcribed genes, RNA-PL having 35% and RNA-PS having 32%. None of the transcribed sequences matched known NCLDV members on NCBI and were excluded from further analysis. Transcribed MCP genes were the third most abundant in the metatranscriptomics at 18%, confirming high environmental expression. Transcribed MCP genes were similar to known NCLDV families and clustered closely to the related metagenomic sequence within the phylogenetic tree, and were closely related and clustered on shared phylogenetic tree nodes, even though they originated from different metagenomes or genomes. Genes from red snow samples MG12 and MG3 were 97% identical, and genes from the environmental red snow sample MG28 and Chloromonas ramsay 005-99 or 047-99 were up to 84% identical. Dark ice samples MG32, MG31, MG19, MG3, and the metavirome each contain a Pol B sequence with greater than 99.5% percentage of identity. Red snow samples MG12 and MG13 from the Greenland ice sheet contain Pol B genes that were greater than 94% identical. Their RNA-C genes from dark ice samples MG32, MG31, MG19, and the metavirome were greater than 99.7% identical, suggesting a degree of relatedness with NCLDVs identified at each sample site. The eukaryotic diversity and composition in all locations are composed of the same members overall, which mirrors the NCLDV marker genes. Interestingly, D5 marker genes from red snow samples and 2 of the algae genomic assemblies clustered closely with giant endogenous viral elements, GEVEs, previously found in diverse green algal genomes. The co-clustering of marker genes found in snow algae genomic assemblies with the red snow sample and GEVEs and other green algae suggests that chlorophytes might serve as hosts in icy environments and contain endogenized viral genes. 10 GV-MAGs and 29 Pol B sequences were obtained from the 31 individual samples. 7 GV-MAGs and 22 Pol B sequences came from the environment. 3 G-MAGs and 7 Pol B sequences came from the algal genome assemblies. I want to note that Pol B is the only marker gene typically found as a single copy and was used for phylogenetic placement within known NCLDV families. In red snow and dark ice samples, a few Pol B sequences had similar or identical residues. Between the 10 GV-MAGs, 3 were 8 unique genome pairs with greater than 99.1% average nucleotide identity and were found in the red snow samples from the Mikdavak Glacier, samples MG12 and MG3, and 2 Chloromonas ramsay cultures 0599 and 4799 indicate a link between giant viruses to red snow algae. There were 5 MAGs that had less than 5 NCLDV marker genes, smaller than 100 kilobase pair genomes, and were eliminated from further study. These MAGs were present in the cryoconite sample, green and red snow, and dark ice. This poses an interesting question. were these potentially unclassified NCLDVs or some other type of ubiquitous environmental virus? The functional annotation of the 10 GMAG showed the presence of genes associated with eukaryotic photosynthesis. The annotations were found within algavirus and 3 astrovirals GMAGs. These genes are often found endogenized in host genomes. The presence of these GMAGs in the environmental snow samples is indicative of potential host-viral relationship. Individual trees of marker genes were built to examine the phylogenetic relationships and diversity between proteins found in metagenomic, metatranscriptomic, metaviromic, and genetic contigs in comparison with known viruses. The maximum likelihood phylogenetic tree of NCLDV marker gene DNA polymerase POL-B clustered in 4 known viral families: Alomenaviridae, Pithoviridae, Asaforaviridae, Algavirus AGO4. There was clear separation in the NCLDV groups by sample type. POL-B from the red snow, N is 3, and green snow samples, N1, grouped together with Aspergillus reference sequences, which is a globally distributed group known to infect phylosynthetic dinoflagellates as well as protozoans. The rest of the red snow, N4, and sequences from snow algae assemblies, C. ramsii, N3, and Microglena, CF species, N1, clustered with the Amenivirus reference sequences, which is the widest order infecting a variety of hosts, including green algae. Signatures found in green snow samples, MG27 and MG26, formed a sister group with Heterosigma Akashiwu virus 01, a microbiological agent for red tide control. Sequences from dark ice were assigned to Pithoviridae, which usually infect the amoebozoan genus Acanthamoeba. POL-B phylogeny shows a wide diversity of NCLDVs and reveals potential top-down interactions affecting a diverse eukaryotic host community on the ice sheet. Red snow, MG28, MG12, and MG3 contained NCLDV signatures of different families. MG28, along with snow algae GVMAGs, were assigned to the family Alomenaviridae. GVMAG MG28 MD2 and Chloromonas ramaceae 005-99 fell within a cluster formed by TET-V. One green snow GVMAG MG27 was assigned to the family IM18 of the Amenivirus order. And interestingly, this family is represented only by genomes derived from cultivation-independent approaches from freshwater and marine sources, and there are no isolated members. One red snow GVMAG was assigned to the order Algavirus. 3 red snow GVMAGs from the Mikdavak Glacier were assigned to the Asphovirus family AF2, and 1 was assigned to a cluster with unknown taxonomy. Generally, this family infect a mix of metazoan and protist hosts and are ubiquitous in marine environments. The presence of Pithoviridae signatures in dark ice and the likely association with protists suggests that the GVMAGs from red snow assigned to the Aspergillus are also probably associated with protist hosts. The active 18S ribosomal RNA eukaryotic community consisted of various algal and protistan members. The variety of active algae and protists in different Greenland ice sheet habitats that contain NCLDV marker genes suggests that infection can occur on a range of eukaryotic hosts in icy habitats. Dark ice was dominated by phylum Streptophyta, mainly from the class Zygomatophyceae at 7 to 37%, chlorophytes from classes Chlorophyceae 5 to 21%, Trebbiophyceae 9 to 27%, and the Cerazoan Glycomonodida at 0 to 1%. One dark ice sample, MG32, was used to try to sequence and assemble a draft genome of Streptophyta glacier ice algae, but contained more than Streptophyta contigs, so it was considered as the 19th environmental sample. And one note about this certain sample is that it had different extraction, sequencing, and assembly methods compared to the other metagenomic samples. Green snow contained algae mainly from the phylum Chlorophyta at 20 to 22%, the protist Cerazoan Glycomonodida at 4 to 10%, and ciliate Stoecksia. Red snow contained algae from the phylum Chlorophyta at 28 to 69%, but the protist Cerazoan Glycomonodida at 1 to 2%, and the ciliate Stoecksia. An interesting note about Stoecksia is it is a large ciliate that contains endosymbiotic green algae and is found in spring phytoplankton blooms and oligomesotrophic lakes, but was present and active in a dark ice sample from the Heim Glacier, MG19, at 33%. The presence of active members of the community in all sample types was confirmed by read recruitment analysis. Reads on the metagenomic samples recruit to the corresponding metatranscriptomic sample. Most reads from each sample map to their respective assemblies, but also map to other environmental types. 10 GV mags, 23 metagenomes, 1 metavirome, and 18 metatranscriptomes. So this paper did demonstrate that NCLDVs were present in almost all of the samples, and they were also present in all Greenland ice sheet habitats sampled. The highest abundances were recovered from red snow and snow algae genomic assemblies, followed by green snow and dark ice. Metagenomic data from red and green snow contain evidence of giant virus metagenome assembled genomes from the orders Emitiviralis, Asfiviralis, and Algaviralis. Metagenomic evidence reveals diverse NCLDV signature genes in snow and ice habitats, indicating the potential of viral controls on these algal communities. Further studies need to be done to measure NCLDV's influence on the snow and ice algal community, as well as the potential to serve as regulators of algal blooms. If further studies do indicate that NCLDVs can control these algal blooms, it could potentially slow ice and glacier melt, which in turn would have global impact on plants, animals, weather, commerce, and coastal erosion, which ultimately affects the food supply. So a couple of questions. Did the sample locations and size affect the types of algae recovered in the environmental samples? Did the stringent QC potentially limit the detection of novel NCLDVs that could control algal blooms more efficiently than known NCLDVs? Is there any significance in the absence of NCLDV viral signatures in the ice core sample? What were the researchers trying to accomplish by leaving out the 5 mags with less than 5 NCLDV marker genes and smaller than 100 kilobase pairs? And why was the dark ice sample MG32 processed using different extraction, sequence, and assembly methods than the other samples? And why did they still include it in the study? And here are my references, and I appreciate your patience. This was a very information-dense paper, and I tried to squeeze it all in 35 minutes, but I appreciate you listening.